okay?
Yes.
Okay, if you cannot hear me very well at any time, just let me know, and I'll try to get closer to this mic. Last year, when I gave this lecture, I had strep throat. So it was like the worst public speaking event I've ever done. But this year, you guys only have to contend with the tail end of a cold. So I'm just cursed to always be sick whenever I get this lecture. But if you can't hear me, just let me know and I'll lean it close. So today we're going to talk about the limbic system and the hypothalamus. And a lot of what we're going to go over, you guys have already seen before. A lot of the structures you've already identified. So this is more of just putting things together, looking at the pathways and kind of how everything works to achieve different behaviors and functions. So first we'll start with our mystery pathology. In this case, an 18-year-old male presented with acute onset of fever, vomiting, confusion, and severe headaches that lasted more than 10 days. At clinical examination, he would not maintain eye contact and he wouldn't respond to verbal commands. He was disoriented, but he had no sensory or motor abnormalities, but he did have severe global aphasia. What is global aphasia? Before we continue. I heard somewhere over here.
Difficulty speaking.
So it's difficulty speaking and understanding language. It's global aphasia. MRI showed changes in the temporal lobe. You can see there's kind of something going on over here with this temporal lobe. And CFS was positive for herpes simplex virus type 1. So he was treated for herpes encephalitis with the appropriate medical treatment for 10 days and he was discharged. So happy ending, right? Well, a few weeks after discharge, he developed some very strange behavioral abnormalities. He would make indecent gestures to women in the street. That also included female family members. He would use abusive language, and he had this new habit of just putting his hands down his pants. So he was kind of an obscene individual after this event. He also exhibited a diminished fear response and unusually low aggression to others in spite of being beaten up by them because of his indecent gestures in the street. He was getting beat up. He didn't seem to care. His appetite increased, and he would steal food off of other people's plates. And lastly, he could not recognize familiar household objects and would kind of have to like play with them in his fingers before he could identify them. So as we're going through the lecture, keep this example in mind. Reflect on the facts of the case, what you know and what you don't know, and how this case could relate to today's class. All right, so let's start off with the limbic system. So there's really two main divisions in the limbic system. There's the limbic lobe and then various subcortical nuclei. Now in the limbic lobe, we have the cingulate gyrus. You guys have already identified that. The prefrontal cortex, the subcallosal area, parahippocampal gyrus with the hippocampal formation, and the uncus. And in our subcortical nuclei, there's the amygdala, the nucleus accumbens, septal nuclei, thalamus, and hypothalamus. Now you'll notice that some of these are color-coded. So this is because some of these structures are intimately related to each other. So we'll talk a little bit more about that in later slides. But the subcallosal area and the nucleus accumbens and septal nuclei are very intimately connected, as are parahippocampal gyrus and hippocampal formation, as well as the uncus and the amygdala. So that's why those colors are there, just to show you that these kind of go together. So first talking about limbic lobe structures, like I said, you guys have seen a lot of this already. So here we've got a little diagram of a mid-sagittal section, not completely mid-sagittal, but a sagittal section. Got the cingulate gyrus up here, prefrontal cortex, of course, is the very front of the frontal lobe. The subcallosal area, and why do you think it's called the subcallosal area? Because it's below the corpus callosum, exactly. That's housed here, below the corpus callosum. And parahippocampal gyrus, which is kind of most easily seen on the ventral surface of the brain. We have an example of here. Now here we've got the uncus, which is kind of this little end pointed part, and then the parahippocampal gyrus coming right off of it. And we've got a couple different colors here in hashed lines. The red part here is representative of the piriform cortex, and the green is in the entorhinal cortex. And these are both very important inputs to the limbic system. Now, what is housed in the piriform cortex? Any guesses? I don't remember if we talked about this before. But primary olfactory cortex is in the piriform cortex. That's where primary olfactory cortex lives. Now, why is the olfactory cortex right next to all of this kind of like memory, emotion stuff? Why do you think that is? Yeah, so smell and memory are very intimately connected. And I have a very recent example of this that's probably too much information, but I'm going to tell you anyway because it'll help you remember. So while I was sick over the weekend, I was in this kind of delirious state, and I just decided to watch one of my favorite shows, to like guilty binge, and that's Ghost Adventures. Has anyone ever seen Ghost Adventures? It's like the dumbest show ever, but I love it. It's like a bunch of bros go into haunted houses and yell at ghosts for an hour. It's hysterical. But because I was so sick, the only thing I could really smell for like a good 4 days was just like my own snot. And you know, like when you're sick, it's kind of that disgusting, weird smell. So I was just that while watching Ghost Adventures. And for the last like 3 nights while I tried to go to sleep, because I'm still smelling that smell, I'm getting flashbacks to Ghost Adventures and like getting scared as I'm trying to fall asleep. So I'm like laying there, I get this like intense smell memory. It's just like there's ghosts everywhere. But that's because smell and memory are so intimately connected with each other. It's like when you smell a certain like, apple pie smell, you might think of the apple pies your grandma used to make or something like that. Or you might think of Ghost Adventures while you're sick. Take it. So we've got entorhinal cortex here and the piriform cortex here on the nucleus. Now, hippocampus, this is the big limbic lobe structure. You guys have already identified it before, but whenever you want to find the hippocampus, look for the cinnamon bun. And so where is it here? Don't look at the labels. Where's the cinnamon bun? Right there and right there. So whenever you have this kind of coiled looking structure, that's probably going to be the hippocampus. So this is our coronal view. But even when you have kind of weird cuts like this, cinnamon bun is right there. And actually, yep, right there. So it's kind of rolling onto each other like a cinnamon bun. So whenever you see that, you know it's probably hippocampus. Easy to identify structure. But keep in mind that this is another one of those circular shaped structures. So what's another kind of circular structure that we have in the brain? It's like this giant C shape. Corpus callosum.
Caudate.
The caudate, yes. So kind of figuring out how these things orient together with the caudate and the hippocampus can be a little bit confusing. But just know that, so this is a dorsal view looking down from the brain. And it kind of cut away some of the cortex to reveal this C-shaped hippocampus and how it kind of starts here and curves up and goes anterior. But remember that the structure is medial to the temporal horn of the lateral ventricle. Okay, lateral ventricle, temporal horn. And then lateral to the caudate. So here we've got a couple of diagrams. This is showing the ventricles. Again, another big C-shaped structure with the lateral ventricle. The caudate is here, with the head of the caudate starting more anterior and then extending posteriorly and then ventral. And then the hippocampus is going to be lateral to that. Yes?
Here, there on the lateral ventricle, we have anterior, interventricular body, atrium, posterior, and the inferior horn.
Inferior and temporal horn are the same. Yeah, in case there was any confusion. Got a little diagram, or a little model to show what your hippocampus might look like in your head. So hippocampus is this giant C-shaped thing with the amygdala sitting right on top. The caudate is going to be kind of tucked in here in its giant C shape. Does that make sense to everyone? So when you go up to lab and you're kind of looking at the different sections, just keep in mind the 3D orientation of these two structures, because they can get easy to confuse. Okay, so why does the hippocampus look like a cinnamon bun? It's kind of a weird, interesting feature. And that's because actually very old cortex that's all rolled up in itself. So when we've talked about cortex and the sulcine gyri before, we've mostly focused on what's called neocortex or isocortex. And that just means that it's got 6 cell layers. So did we talk about cell layers, the different layers of cortex before? I don't think we covered that. Don't worry about that, just know that there's 6 layers. And in terms of the phylogenetic age of this neocortex, it's pretty new. And that includes motor cortex, somatosensory cortex, visual cortex, all of those. There's also paleocortex, which has 3 to 5 cell layers. It's a little bit older than neocortex in terms of phylogenetic age. And examples of that are entorhinal cortex and the piriform cortex. And the last thing, we have archaic cortex, which is only 3 layers. And that's real old compared to these two. You don't need to know exact numbers, just know it's real old. And example of that is the hippocampus. And just real quick, these two combined are known as allocortex, which just means cortex that's fewer than 6 cell layers. Does that make sense to everyone? So in terms of phylogenetic age, we started out with 3 layers, and then we worked our way up to 6 to neocortex. All right, this is a cross-section of the hippocampus. You see the cinnamon bun. And that cinnamon bun is composed of 2 C's. Got the little c here, and then the big C here. Now that little c is the dentate gyrus. The big C is called the cornu-ammonis, or CA region. And we'll talk about the functional distinctions of these different regions in a second. But the little c is dentate gyrus, big C is cornu-ammonis, or CA. Coming off CA, we have the subiculum. Okay, subiculum. And then coming off that, we have a structure called the alveus, which is a bundle of axons coming off the subiculum and wrapping its way back around. Because remember, everything is kind of rolled together in this structure. And then this bundle of axons, the alveus, turns into the fimbria, and the fimbria then becomes the fornix. So dentate gyrus, CA, subiculum, alveus, and fimbria. And then our big input to the hippocampus is the entorhinal cortex on the parahippocampal gyrus. Now, in terms of how information flows through this structure and the different functional components of the structure, our major input, like I said, is the entorhinal cortex. And the projection from the entorhinal cortex to the dentate gyrus is known as the perforant pathway. Perforant pathway is entorhinal cortex to dentate gyrus. Now from the dentate gyrus, the projection flow of information then goes to the CA region, which is here. And then from CA to subiculum to alveus to fimbria to fornix. So this is the flow of information over here. And our major output is the fornix. Now, we mentioned that this structure has 3 cell layers, mainly 3 cell layers. And that's composed of afferents into the structure, a layer of cell bodies, and then a layer of efferents out of the structure. So in the dentate gyrus specifically, the molecular layer is composed of afferents into the dentate gyrus. So that's going to be axons coming from the perforant pathway, as well as dendrites from neurons that live in the dentate gyrus. The granule cell layer is the layer of cell bodies, and then the polymorphic layer is the layer of axons projecting to the CA region. Now when we get to the CA region, which is sometimes called the hippocampus proper, but it's technically the CA region. That's why it says hippocampus here. Again, the molecular layer is those afferents coming into the CA region. So those are projections from the dentate gyrus. The pyramidal layer, those are the cell bodies of the CA region. And then again, the polymorphic layer are the efferent axons then going to the subiculum and through the alveus and the fibria. So just remember, we've got 3 layers, the afferents, the cell bodies, and the efferents. And really the only difference is the name of the cell body layer. In dentate gyrus, it's the granule cells, and in hippocampus, or in the CA region, it's pyramidal cells. Now this figure over here is showing the different subsections of the CA region, and these have their own special circuitry as well. So from the dentate gyrus, we have projections going to what's called CA4. We have projections to CA3, CA2, and then CA1. So it kind of starts with the biggest number and then goes around. We start with 4, then go 3, 2, 1. And then the subiculum would be over here. There's also a projection that's really talked about a lot from CA3 to CA1 called the Schaefer collaterals. And those are very important in studies of memory and the limbic system. These are these are the inner circuitry of the hippocampus. So from dentate gyrus, we go to CA4, CA3, CA2, CA1, and subiculum. They're also all kind of interconnected with each other too. Okay, so we talked a little bit about memory, but let's get a little bit more specific. So the hippocampus is important for not memory storage, because that occurs in other cortical areas, but it's important for consolidating short-term memory to long-term memory. So here we have an example of a normal brain and a brain from, well, these are both diagrams, but example of a normal brain and then a brain from HM. Does anyone know who HM is? He's a really famous case in neurology, HM. He died a few years ago and they released his actual name. It's like Henry, what is it? Dorson? Yeah. So HM was a guy, I think he had temporal lobe epilepsy, and they would originate in the hippocampus. This was before we knew that the hippocampus was really important for memory. So some surgeons were like, well, you're getting seizures here, let's just take the structure out and see what happens. So he had a bilateral removal of the hippocampus. And after that, he kept his long-term memory. So like the memories he had when he was a kid, still had those. But he couldn't form new memories. So he kind of had to live moment to moment. And he was studied for a very long time. And this was kind of the first time that we learned that the hippocampus was really important for this memory consolidation. And how that happens is extremely complicated and not completely understood. But we do know that there's these 2 phenomena called long-term potentiation and long-term depression. And for the purposes of this class, all you really need to know is that long-term potentiation is the strengthening of synapses in these different regions, CA regions and the dentate gyrus, while long-term depression is the weakening of synapses. And this doesn't just occur in the hippocampus, it also occurs in the amygdala, the cerebellum, and a lot of other structures. But in terms of this whole short-term to long-term memory consolidation, long-term potentiation and long-term depression are really important for this whole process. Are there any questions before we move on to other structures in the limbic lobe? So let's talk a little bit about the different subcortical structures. We'll start with the big one, which is the amygdala. And again, this is something that you guys have already identified. So here we have a coronal view. The amygdala is this collection of cell bodies right here. And this is going to be anterior to the hippocampus. Here we have excuse me, an axial section, and the amygdala is right here. On the other side. Now there's 2 major divisions of the amygdala. There is the basolateral group of nuclei, which has a lot of cortical connections. And then there is the corticomedial group, which is very heavily involved in olfaction. There's olfaction coming back again. Now, function of the amygdala has a lot of different roles. It's involved in emotional learning, and particularly fear memory, but it's also involved in the modulation of long-term memory. and particularly emotional memories. So one paradigm that has been used to study this is something called fear conditioning, which is basically you take, well, you can do it with people too. So you take a person and you give them a mild shock, pair it with a particular tone, and then after a few times of that happening, when they hear that tone again, they have a fear response because they're expecting the shock. So they've learned that the tone goes with the shock. It's all classical conditioning. But when you take out the amygdala, they no longer have that fear response, and their fear learning is severely impaired. All right, other subcortical limbic structures. We've got the nucleus accumbens and the septal nuclei. And these are both anterior to the amygdala and the fornix, and they're kind of housed in that subcolossal area that we pointed out before. So in this coronal section, the nucleus accumbens is right here. These little spots here. And the septal nuclei are here. So you'll notice these kind of familiar looking structures. So what is this dark band here? Internal capsule. And what's this? Putamen. And what's this? Cotton. Yes. So, yes.
Which limb of the internal capsule is that?
This would be the interior one, because we're really far forward, really far interior. So one thing to keep in mind when you're trying to identify this structure is one, you have to be really far anterior. So anterior that the internal capsule kind of stops right here, and it looks like the caudate and the putamen are kind of like joined together, but they're not. That's the nucleus accumbens. That's going to be that little spot right there where the internal capsule kind of stops. So when you see something like this, you know you got nucleus accumbens. Now these two areas are implicated in emotional processing, emotional memory, reward, and especially addiction. A lot of people at Wake study these areas of the brain in terms of their role in addiction. And just to quickly mention this, both of these structures are really intimately connected with this area called the ventral tegmental area in the midbrain. We're not going to ask you to identify that. Just know that the connections between these two areas are very involved in addiction. Okay, so talking about projections. Here I have color-coded a few things to try to make it a little bit easier to keep everything straight. So our referent structure is in blue, and our afferent structure is going to be in green. So afferents to the hippocampus, the main one is going to be the perforant pathway, which is a whole bunch of incoming cortical input into the hippocampus. And that's mainly coming from the entorhinal cortex. In terms of the amygdala, for the basolateral cell group, we have afferents from the thalamus, other parts of the limbic lobe, and other cortical regions. Pretty big. We're not going to get specific for this. For the corticomedial cell group, we've got olfactory inputs from the hypothalamus, the thalamus, as well as brainstem nuclei. So remember how the amygdala is involved in kind of the fear response. We've got all this brainstem input, both to the amygdala and from the amygdala. And you can kind of think how, yeah, you need to get your heart rate up, you need to get your breathing going, it's all part of the fear response. So it's going to be connected with the amygdala. In terms of nucleus accumbens and the septal nuclei, we have afferents from the fornix. These two pathways, we'll talk about in a little bit, the stria terminalis and the amygdalofugal pathway, as well as, like I said, the ventral tegmental area. Efferents from the limbic system. From the hippocampus, the main one is going to be the fornix, and that projects from the hippocampus to the mammillary bodies and the septal nuclei in particular. And I want to point out that there's this split that happens with the fornix. So we have the pre-commissural fornix and the post-commissural fornix. The commissure part is referring to the anterior commissure, which is that projection connecting the two hemispheres. It's very interior to everything. So the fornix goes in front or anterior to the anterior commissure, and with that, it's projecting to the septal nuclei. And then the post-commissural fornix is projecting to the mammillary bodies. And I've got a couple diagrams on here to illustrate that. Now, these two pathways are both coming from the amygdala. The stria terminalis goes to the hypothalamus, the nucleus accumbens, the septal nuclei, as well as the caudate and putamen. The amygdala-fugal pathway goes from the amygdala to the hypothalamus, septal nuclei, cortical areas, and again, brainstem areas. There's also the medial forebrain bundle, which contains both afferent and efferent fibers between the septal nuclei and the nucleus accumbens, and then the hypothalamus and the brainstem. Okay, here we've got another coronal section to just show you the location of a couple of these things. So you've already identified the fornix. This is part of the fornix. It's kind of very medial structure coming down. And then the stria terminalis, which is that structure going from the amygdala to all these different places, is actually medial to the caudate. So this is another structure, a projection where we've got this kind of C shape happening from the amygdala, projecting in a C and then going anterior. So when you see the caudate and kind of this little, this is the terminal vein, when you see that, and the stria terminalis is right there. So on the other side. Medial to the caudate. And the amygdala-fugal pathway is difficult to find, so don't worry. Okay. The last kind of circuit we're going to talk about with the limbic system is known as the PEZ circuit, and this is involved in memory as well as emotional regulation. And first, let's just go through this kind of, it's a cycle, you know, it's a circuit. We'll just go through the little diagram first, and then we'll look more at the anatomy in a second. Basically, we're getting input to the entorhinal cortex, which is then going to go via the perforate pathway to the dentate gyrus, and then we have all of these hippocampal connections from CA3 to CA1 to the subiculum. And then via the fornix, that's projecting to the mammillary bodies in the hypothalamus. From the mammillary bodies, we then go dorsal to the anterior nucleus of the thalamus. And that projection is called the mammillothalamic tract. Mammillothalamic tract. So again, it's in the name, mammillary bodies to thalamus. From the anterior nucleus of the thalamus, we go to the cingulate gyrus. And then, let's just ignore this part, from the cingulate gyrus back to the entorhinal cortex, and there's this loop. And that is involved in emotional regulation and memory. So if any of these are kind of taken out, then you have impairments in emotional regulation and memory. So in terms of the anatomy and kind of putting it together in 3D space, here we've got entorhinal cortex projecting to the hippocampus with all those hippocampal connections. And then from the fornix, we have C-shaped fornix projecting anteriorly. And here we can see the split between the pre- and post-commissural fornix. So here, this little dot is representing the anterior commissure. And here we've got the pre-commissural fornix going to the septal nuclei and the nucleus accumbens, and the post-commissural fornix going down to the mammillary bodies. From the mammillary bodies, we go up to the thalamus, to the anterior nucleus, via the mammillothalamic tract. And we go back up to the cingulate gyrus, and then the whole cycle starts again. Everyone clear on that? So, given that you guys are all experts now in the vasculature of the nervous system, I'm going to ask you, what is your best guess as to what supplies blood to the limbic system? then ACA here, and what is that supplying? Cingulate gyrus, as well as these subcolossal areas. And then this is just your ventral view showing the PCA projected back. Here it's going to also innervate the uncus and the parahippocampal gyrus. All right, now we're going to switch gears to the hypothalamus. Here we've got our mid-sagittal view, thalamus here, and then right ventral to it is the hypothalamus, this kind of triangle-shaped structure. Now, the hypothalamus is divided into a couple of different zones. Here, this is a representation of kind of a top-down view, so like you were looking down dorsally. The most lateral part is the lateral zone. It's easy to remember. We then have the medial zone, which has a lot of the different hypothalamic nuclei in it. And then the paraventricular zone, which is right next to the third ventricle, which is depicted here. Other subdivisions are the supraoptic region, which contains these nuclei in green here. The tuberal region, which is the red nuclei. And then the mammillary region, which is kind of gray looking nuclei back here. Okay, so we're not going to make you learn all of the nuclei, which is lucky for you, but you will need to know some. So first, I just want to say that the lateral zone contains the lateral nucleus as well as fibers from the medial forebrain bundle. Other nuclei, we have the suprachiasmatic nucleus, which is in the supraoptic zone, very medially. The paraventricular nucleus, which is the nucleus of the paraventricular zone. The paraventricular nucleus, so peri versus para, don't get them confused. Paraventricular nucleus, which is in the supraoptic region. The arcuate nucleus in the tuberal region, as well as the dorsal medial nucleus. And then, of course, we have the mammillary body back here. We also have the supraoptic nucleus located here. And I just want to say before we go any further that we're not going to really expect you guys to look at the gross specimens and say, oh yeah, this is where the supraoptic nucleus is. That would be really challenging. But know how they are situated. We might ask you for one or two in micrograph. I'm not sure yet, but just know how they're oriented and how they're situated. And we're going to go through a few examples of different micrographs so you can see some of the different nuclei. Some of the more easily identifiable ones. So we have the supraoptic nucleus, and the ventromedial nucleus is also in the tuberal region. Now we have a mid-sagittal view. So this is our little triangle structure. Again, there's a supraoptic region. Excuse me, there's a mammillary region back here, the tuberal region in the middle. Just pointing out these different nuclei again, and we'll go over the function of these different nuclei. That is probably the most important thing for you to get out of this. So the suprachiasmatic nucleus, very anterior. The supraoptic nucleus, which also right next to it. The arcuate nucleus in the tuberal region. Ventromedial nucleus right above it. The paraventricular nucleus here. And our mammillary bodies here. Okay, so here is a few examples of a coronal view showing some of these different nuclei, how they would actually look, not just a diagram. All right, so we've got a few different cuts, A, B, and C. And you'll notice that this is going through the supraoptic region, this is going through the tuberal region, and this is going through the mammillary region. So for A, this is our most anterior portion. You can see the optic tract, the optic chiasm here. We see the anterior commissure here. Oops. And we've got the supraoptic nucleus, which is located right next to that optic tract. Let's see here. The anterior nucleus, we don't go over that one. Actually, I think that's it for the ones that we want you guys to know in this one. In B, this will be the tuberal region. You see the optic tract here again. So keep in mind, this is optic chiasm, and this is optic tract now. We've got the dorsal medial nucleus, very medial, next to third ventricle. The supraoptic nucleus, the ventromedial nucleus, and the arcuate nucleus. So they all kind of line up together. And then our paraventricular nuclei, those are kind of easy. Those are just right next to the paraventricular zone. C, this one is pretty straightforward. This is our mammillary region. We've got our mammillary bodies right there. Third ventricle here. Now in terms of function, these are the nuclei that you really need to know kind of the function of. So we've got the suprachiasmatic nucleus. Now when this is stimulated, it adjusts your circadian rhythm, your circadian clock. And when it's abolished, you get abolishment of the circadian rhythms. So your whole sleep-wake cycle is thrown off. The supraoptic and paraventricular nuclei are involved in blood pressure and blood volume. And lesion of this results in diabetes insipidus. Right. The lateral hypothalamic nucleus is involved in feeding behavior, and when it is lesioned, you have decreased feeding, or anorexia. The ventromedial nucleus is also involved in feeding behavior, but when it is lesioned, you have increased feeding. The dorsomedial nucleus is involved in emotion and aggression, and when it's lesioned, you have a decrease in aggression and just decreased affect altogether. And the mammillary bodies are pretty interesting. Stimulation, we've got a question mark there, but when this is lesion, short-term memory is not processed into long-term memory. Now, why would that be? What circuit did we talk about a couple of minutes ago that involved mammillary bodies? The Pepys circuit. Yeah, it's one of the stops in that circuit. So when you cut that out, you're going to have problems with your short-term memory and long-term memory. Now, hippocampal afferents. We're getting input from the fornix, from the medial forebrain bundle, the stria terminalis, and then the amygdalofugal pathway. So these are all structures we learned about before. Efferents from the hypothalamus, there are a lot. We're just going to talk about these three, though. We have the mammillothalamic tract, and where is that going? Right, the anterior nucleus of the thalamus, mammillothalamic. And then we've got these two kind of internal tracts in the hypothalamus. The supraoptico-hypophyseal tract, that's a mouthful, and then the tubero-infundibular tract, also a mouthful. What are these doing? So the hypothalamus is very intimately connected with the pituitary gland, right? It sits right below it. Here we've got the hypothalamus, and then the pituitary gland sitting down here. And connecting them is the structure called the infundibulum. So it's right there, infundibulum. Or the infundibular stalk, it's also called. And the pituitary gland also has its own special divisions. The anterior lobe is called the adenohypophysis, which is a fancy name for the anterior lobe. The posterior lobe is the neurohypophysis. And we've got nuclei from the hypothalamus projecting to these different parts of the pituitary gland. Now the supraoptico-hypophyseal tract is projecting from a couple of different nuclei, but for this we're just going to focus on the paraventricular nucleus and the supraoptic nucleus, down to the posterior lobe of the pituitary gland, or the neurohypophysis. And this projection is involved in oxytocin secretion. Has anyone ever heard of oxytocin? So what does that do? Any guesses? It's like the love hormone, which is a complete oversimplification. But we've got oxytocin here, as well as ADH, which is, where is it? AHC? Diuretic hormone, yes. So that's a direct projection from the hypothalamus to the pituitary gland. Our other tract, the tuberoinfundibular tract, here we're just going to focus on the arcuate nucleus, but again, there's other nuclei involved. That is projecting from the hypothalamus down into the infundibulum. And here it's going to secrete hormones into the capillaries located here, and then those go down to the anterior lobe of the pituitary, or the adenohypophysis. And so it's kind of not a direct, or it's an indirect way of getting hormones and getting excitation of the pituitary gland. So tuberoinfundibular stops off in the infundibulum, and then the capillaries are carrying these signals down to the anterior lobe. And the anterior lobe is really involved with growth hormones, different growth hormones. Okay. Any questions about these two tracks before we move on? Blood supply to the hypothalamus. Again, we've seen all of these different arteries before. It's mainly going to arise from these small arteries extending from the circle of Willis. So we've got the posterior cerebral artery gives off little artery branches that are going to the mammillary bodies. But the anterior communicating artery, as well as the anterior cerebral artery, giving off little branches here to the hypothalamus, as well as the posterior communicating arteries. Okay, so what about our mystery pathology? This guy had this inflammation, this viral inflammation in his brain that affected a lot of the temporal lobe. And then he had all of these kind of weird behaviors, right? He had kind of this hypersexuality where he was making indecent gestures to women in the street. He had decreased aggression. What structures do you think could be affected?
Amygdala.
Okay, amygdala. Yes. Okay, parts of the hypothalamus, that could be. I heard something else. A little rumble, that moan that you guys do. You don't want to be heard. Okay. What is the diagnosis? That's all right, I didn't expect you guys to. This is an example of Kluver-Bucy syndrome, which is basically just temporal lobe dysfunction. So this can appear after stroke, after temporal lobe surgery, after different infections in the brain that result in a lot of inflammation. It's characterized by amnesia, docility, dietary changes, hyperorality, hypersexuality, visual agnosia, and lack of affect. So here is our patient. We see he's got all of these temporal lobe problems. Includes amygdala, includes probably part of the parahippocampi gyrus, probably part of the hypothalamus. All of these structures are affected. And here we've just got some kind of silly examples. This cat has Kluver-Bucy syndrome, so he's trying to get a little frisky with the dog, which, as we know, he shouldn't be doing, but his temporal lobe's all messed up, so he's not quite sure what's going on. And then here we've got a monkey and a snake, and the monkey has Kluver-Bucy syndrome and has lost his natural fear of snakes. So instead of kind of recoiling from the snake, he's actually reaching out to touch it. And this is a classic case of Kluver-Bucy.